welcome to CFDE Pediatric Power Up. Like I said, my name is David Higgins. I'm the Informatics Program Manager at the Center for Data-Driven Discovery in Biomedicine, which is an organization within the Children's Hospital of Philadelphia. And so today I'm kind of wearing my hat as a trainer to kind of talk about different data sets that we make available through Kids First DRC and our partnership with the Common Fund Data Ecosystem, CFDE. So I'm joined today by my co-instructor, Sangeeta, and so I'll pass the microphone to her and let her do a brief introduction of herself as well.
Hello, everyone. My name is Sangeeta Shukla, and I also assist Dr. David Higgins with this Kids First and CFDE program to enable researchers get more comfortable with pediatric data access and resources and tools around pediatric research. And I'm very happy that you all have joined us today. I'm looking forward to speaking with you all on more topics tomorrow and later.
Absolutely. So thank you so much, Sangeeta. And then we're also joined today by Chris Nemrich, who's one of our project managers. He's helping to organize the course and keep us on track and keep us moving forward. So he may be helping us answer some questions in the chat. So if you see his name pop up there, that is another friendly helping face. So that's the three of us who are going to be kind of the main folks who are running the course this week. So what are we doing? So today is day one of the 5 days. And so I want to provide an introduction to what is this workshop? What are we learning? What are we going to cover? Is this the right thing for you to join and might learn from? Today, we're going to provide an introduction to what is the CFDE, what is this organization, and this concept called DCCs. Talk about different types of data sets that are available, and we're also going to explore methods for accessing data. So we're just kind of a very introductory type of day, and we're going to scale up as the week goes on, as I'll talk a little bit more when I dive into the workshop. So what is our workshop? So the goal for this course is to teach you skills in using Common Fund data sets to support research in pediatric biology. And so that feels very kind of broad and grand, but I really want to focus on one particular word we were trying to focus on is you. So we're trying to do our best that we can to reach folks at all different kinds of levels, wherever you're coming in from, to be able to help you understand a little bit more about what is out there in terms of these genomic data in order to be able to support your research project. So when you signed up for the course, you filled out a brief survey asking you about, what are your skills? What do you know about this? How would you describe your level of comfort with that? So I'm just going to share one answer with you. So one of the questions was, how do you use omics level data as part of your research? And so I provided kind of 4 different options here, from kind of I feel very comfortable, I do this every day, to I have no skills at all and I'm hoping to learn skills, and then kind of a range in the middle. And so we got a very broad set of responses. You can see that it's almost equal in terms of, divided up by quarters of how folks kind of self-identify their own skills in using omics level research. So it's from an instructor standpoint, I think our opportunity here is to really try to offer a course that provides kind of a really broad set of information that can hopefully, provide information for all folks. And no matter where you self-identify on this level of responses here, hopefully you'll be able to take something from our course and use it to support your projects. So here's our outline that we're going to be talking about in our course today. So today we're on day 1. We're going to provide the introduction to the Common Fund data ecosystem, and as I kind of talked about at the top of the lesson today. But the design of the course is that it's really meant to kind of scale up as we go. And I think I mentioned this in an email on Friday, if you had a chance to read through that. So we're starting very kind of basic in terms of what is CFDE, what is the types of data out there, what are the types of things that I can use in my research. So this is, the kind of the foundational day 1. Day 2, we're going to kind of get a little bit more complex. And so this is where we're going to talk about tools and methods in bioinformatics. This is going to be led by Sangeeta, who's going to lead day 2. And so we're going to learn about, okay, so we've identified this data sets on day 1. Now, how do I use it? How do I interact with it? How do I get information and knowledge out of this data? So we're going to learn about tools and methods in bioinformatics. We're going to talk about different types of coding. We're going to provide an introduction to this concept of cloud computing, and we're going to look into how we can combine some of these CFDE data sets together with specific use cases. So it's kind of building on where we started with day 1. Day 3, Wednesday. Now we're continuing to scale up a little bit further. So day 3, we're actually going to do an analysis. So we're going to do an RNA-seq analysis using a software tool called DE-seq2. And we're going to do that in the Chromatica Cloud platform. So I'm going to be demonstrating that as I go on my screen, but folks will have the opportunity to follow along on your own computer as well as we go through the demonstration. So now we're kind of combining the data sets and the tools and methods together. So now we're actually going to execute it and run it ourselves. And on Thursday, again, scaling up, now we're going to be not just using an existing tool, but we're going to be building a new tool. So Sangeeta is going to be leading the Thursday session as well, where she's going to show us how she builds a single cell RNA-seq pipeline in Chromatica. So she's going to be taking the software tool and setting it up for the first time. So it's starting from a new starting point instead of using existing data existing tooling that's already available. And then Friday is kind of the culmination point. So Friday is not going to be, it's going to have a different format where it's going to be much more open-ended. So we're going to have an opportunity to sit and chat with folks who join on Friday. And we're going to talk about how do we connect individuals with the resources to help you take your project to the next level. So again, wherever you're coming in from, we want to help you. So if you're saying, oh, I don't understand, these different, coding languages, we can provide you with resources to learn that a little bit better. Or if you're saying, oh, I think I'm ready to dive into Chromatica myself, we can provide you and point you towards people and resources to help build your own pipeline, for your own research project. So again, really focused on individuals. We want to try to focus on providing this research project for you to take your stuff to the next level. So to reiterate, so the course kind of scales up. So we start at the more basic and we end in the more complex. So if I go back a slide here. So if you're one of these folks down here at the bottom who identifies as, maybe having few or no skills in computational biology, hopefully the first 2 days are going to be very useful and informative because it will teach you about kind of the background and what's out there. And then that can give you kind of a spark to hopefully, learn a little bit more from the end of the course, but then begin to kind of set you up for some self-discovery after the course. And likewise for my folks who are, self-identify up here at the top of the table, hopefully the days at the end might be more empowering and impactful. And so, where we're actually using the tooling, if you're coming in with the skills and the knowledge already, maybe you're ready to dive in and use the tools. So that's what we're saying is that we're trying to help folks, wherever they are and scaling up to reach folks wherever they're coming in from. And then my last comment is that so the days do not, they kind of scale up in complexity, but they don't build on one another. So each day is meant to be independent. Looking ahead and you're thinking that, oh, well, I already know about tools and methods in bioinformatics, then, you can skip Tuesday and you can come back on Wednesday. And so, or if you're, maybe lost by the analysis, maybe you skip Thursday and then you come back on Friday for the help and Q&A session. So every day is a standalone. You don't need to attend all 5 days. And we're trying to make it, as accessible for folks as possible, understanding that everybody has obligations that they need to work on at their own institutions and their own projects. So they may not be able to join a full, 5-day, week-long workshop. So that's kind of my overview of where we are and where we're going and a little bit about what we're going to come up to next. So with that, I'm going to dive into our day 1 topics here. So again, we're talking a lot today about what is the data that's out there and what is this common fund data ecosystem, which is going to set us up for the rest of the course. So let's jump ahead. All right, so step 1, introduction to common fund data ecosystem and data coordinating centers. All right, so we kind of got to start. I like to start very broad and then kind of narrow in my focus. So starting as broad as possible for this one, I think I'm going to start by talking about this organization called the NIH Common Fund. So the NIH is the National Institutes of Health, institutes being plural, the key S there that folks sometimes forget. And so what that means is that it is a number, more than a dozen smaller groups that have all kind of under this umbrella of NIH. They do operate largely independently with some connecting coordination. Part of that coordination is done by this NIH organization called the NIH Common Fund. So this is a kind of a part of NIH that exists to kind of identify projects that might exist in different institutes and kind of bring them and coordinate collaboration together to help them identify innovative research that is expected to make a broad impact. So this is where projects might be across multiple institutes, and we're trying to bring them together to coordinate. So what are some examples of these common fund programs? So the first one I'm going to talk about is this program called GTEx, the Genotype Tissue Expression Program. So what this program is, it's really focused on investigating the relationship of genomic variants and gene expression. And so what this is, it's a catalog of samples of 54 unique tissues from more than 1,000 deceased participants. And it provides genomic information as well as expression information. So I'm actually going to back out of my slides here for a second. And we're going to pull up, if I find my window, here it is. So we're going to pull it up. So we can search NIH GTEx. And so this takes us to an NIH website, it's a Common Fund website, or it tells us more about GTEx. We're not going to read all the details, since this gives us some kind of overview information, but we're going to explore the GTEx portal, which is you can find by clicking this link right here. And it says, yes, we're leaving the website, and that's okay. There we go. So what we got here, so this is the GTEx portal. So this is the way that you access data related to that's collected as part of the GTEx program. And so they have a couple of new projects. They're looking at kind of developmental GTEx as well as non-human primate developmental GTEx. Those are kind of coming soon. The data is not available yet, but it has been funded. But the data set that we're going to talk about and explore a little bit later on Wednesday is this kind of adult GTEx project right here. And so it provides information about the types of data, the types of samples, the number of donors, and the types of tissues that these samples were collected from. So if we go into a couple of different things, so what types of stuff is in here? So you can click on tissues. You can click on, for example, the tissue sampling sites, and it gives you an overview of this is the sampling sites from the people who consented and agreed to donate to the GTEx program. And then you can kind of highlight different tissues if you have a particular interest. So if you are might study maybe brain tumors, and maybe you're interested in different brain tissues, different parts of the brain. If you are a cardiologist, you might be interested in heart tissue or kidney tissue. And so it's different tissues across the human body. We can also explore, so the samples themselves are not available, but the data that has been collected from the samples is available. So for example, we can look at expression. We can do a search in their expression browser. And so if we go to search for a gene ID, and maybe we're interested in a gene called EGFR, which is a receptor involved in signaling cell division. And so that gives you information about relative expression of this gene, which is shown here on the y-axis, and then it shows across the x-axis here. This is all the different tissues in the body that are sampled for this particular data set. So it allows you to really rapidly search for your gene of interest, and then identify expression of EGFR is relatively low in the brain, but it's much higher in the skin. Which makes sense because perhaps our skin is dividing much more rapidly than our brain cells as adults. So I can provide you information about a particular gene. It has more in-depth information as well, especially related to, for example, you can look at specific exons of the genes that might be expressed, particular splicing variants that might be specifically expressed in different tissues. And so it gives you information about the gene of interest that you search for. It also has some documentation to support all their different tooling. And then it also has an opportunity for you to be able to access the data as well. So their data comes in kind of two formats. They have a large amount of open access data. This is things like expression counts and the reference files and histology images that are associated with these data types. So these are all available open access data for anyone to be able to use. They also provide protected access data. If you're able to apply for access and are approved, you can receive access to the underlying genomic files, the BAMs, the CRAMs, the BCFs, things like that. It also allows you to be able to download individual files. So if we look at bulk tissue expression, for example, we can look within specific gene read counts by tissue. And so if we wanted an expression of every gene from every bladder sample, then we could download this file, 1.7 megabytes, and it would give us this very large table of gene expression values. So this is the underlying data that's used to generate that, that box and whisker plot that we just showed on the previous tape. So if you're interested in a specific, type of expression in a specific sample, you can download by specific tissue as well, in addition to the complete data set. So we're going to come back here on Wednesday, and we're going to show how we can pull data out of here and we can combine it in Cavatica when we're doing an analysis. But just wanted to highlight the types of things that are available here with GTEx. So GTEx is this great resource. It's really great, especially if you're looking for like control type data to be able to compare your analyses to. So it's just kind of a great overview of this GTEx resource. All right, so that is one of our DCCs. So it's a Common Fund program that's funded by this Common Fund organization to produce this resource that's available for investigators across different conditions. The next one I'm going to talk about is Gabrielle Miller Kids First. So Gabrielle Miller Kids First is really important to me because I help work on the Kids First Data Resource Center. So our goal is to help researchers identify new insights into the underlying biology of pediatric cancer and structural birth defects, especially related to finding shared genetic pathways that might exist between these disorders. And so what we provide investigators is a collection of 35 different studies. Each of these have genomic data on a collection of pediatric patients with shared clinical conditions, and then we allow users to be able to access and combine those data sets together. So let's look at this one a little bit more in depth. So I'm going to go back out to my internet window right here. And we can do a search for NIH Kids First in our Google, and it will take us again to the Common Fund website. So this is the NIH organization talking about Gabrielle Miller and Pediatric Research Kids First. But if I want to learn more about the data resource itself, so that's built by the Gabrielle Miller Kids First data resource. So we can click on their website right here, which again, so now we're exiting the NIH website, and so now we're in the Kids First portal. So we've got a website as well, which talks a lot about our projects and our data sets and what we make available to investigators. But the data itself is inside this portal, so we can log in and explore the portal. So to log in, you can you go to this portal website and click the login button. You can log in with any Google ID, any Google authenticated email address. If you have an ORC ID, you can log in with that. If you have an NIH ERA Commons ID, you can log in with that as well, which is what I'm going to log in with today. I'm going to sign in. I'm going to grant permission. And now I'm into the portal. So we're going to go in deeper depth with each of these sites as we go on throughout the week, but I'll just show some of the highlight features here on Monday. So in our studies tab, provides information about different Kids First studies. So right now we have 35 studies on the Kids First portal. And you can narrow these according to specific interests. So if you're just interested in birth defects, you can check the birth defect box. And now we're just looking at those. And so it helps you identify and hone in on different data sets that you might be of interest. So we have a very broad set of data. Like I said, we have 35 studies from head to toe. So we have our Children's Brain Tumor Network study, which is a study of pediatric central nervous system tumors. We have other very large studies. I guess we can sort by participants. Our congenital heart defect study, the PCGC study is also very large. We also have a very big heart defects and lymphoblastic leukemia in children with Down syndrome. So children with Down syndrome are more likely to be diagnosed with heart defects and leukemia. So it's kind of identifying that intersection there. We also have a very large congenital diaphragmatic hernia. And kind of across multiple different studies, we have a number of craniofacial development and orofacial clefting studies as well. How diverse are these data sets? Great question, Harry. So we can explore that on the next tab by clicking Data Exploration, which provides information about, a little bit deeper information about who these participants are. So if we click over to Data Exploration, So this provides information about all the participants in our study. And so we have 34,000 participants, and provides, in terms of who the participants are, we've got this demographic box right up top. So in terms of race and ethnicity, the participants come from research projects that were begun at hospitals, mostly in the United States. And so in terms of the ethnicity of these individuals, they are mostly not Hispanic or Latino. And in terms of race, they're mostly white, but we do have specific information about participants who might have more than one race or have more than one ethnicity. You can even narrow, so if you're interested in particular subset, you can apply filters as well. We also have some studies, especially related to orofacial clefting, that are specific to groups that are specific demographic groups. So we have orofacial clefts for groups of African ancestry. We have a microtia study for participants of Latino ancestry. And so there are some studies that are specifically targeted at identifying conditions in specific populations. So our data exploration tab provides some information about the different study participants as an overview, but we can also narrow down a little bit further. So if we want to identify participants with a particular condition, so we could look for under observed phenotype, maybe we're interested in oral clefting. And so we can select that and we can apply it. So this gives us information. So we have 1,529 participants that have data that are tagged with the ontology code oral cleft. And so then we can scroll through. If we click the participants tab, provides us more information about who these particular participants are. Allows us to be able to explore further. So let's see if I reset my columns here. Here we go. Then it gives us information, about their diagnosis and about their conditions as well. Question from Harun asks, are these data sets only diagnosed ones, or do they also include control groups? So not all participants have the condition. So we also include trios. So for example, this participant here can pull up, let's see here. We pull up this participant's family. So we have trio. A lot of these studies have trios. So we have the proband child who's diagnosed with cleft lip. And then we have this participant's mother and father who do not have cleft lip. And so you can use this to identify, so you could use the parents in this way. But Kids First is really focused on overall participants who have the condition. So we don't have any control groups or control data sets, but this is where data from other NIH Common Fund programs like GTEx can come in. And so that's kind of the crux of our course, is that if we combine these different data sets together, it empowers us to make even stronger discoveries. So yes and no. There is most of the participants we're focused on are going to have the one particular condition of interest as well. All right, and so then from there you can identify the data files that are associated with these participants. So we have some alignments in here. We have some variant files, and we have a joint called family VCF as well for information about this particular family. All right, so that's kind of Kids First in a nutshell. And I'll show one more feature about Kids First. So Kids First also we've recently launched this variant explorer tool. So what this does is it allows you to be able to search for variants in the Kids First data without necessarily having controlled access to the controlled access genomes. And so again, you can go back to, if we go to our variants tab, and if we have a gene, what's our favorite gene? Maybe it might be, we'll go with BRAF. Nope, I'm in the wrong section here. If we go to gene, and then we type BRAF, then we can look at, these are all of our participants in the Kids First Data Resource Center data sets that have mutations in BRAF. And so it's sorted based on deleterious consequences. So these are, affecting, early stop codons. These are missense mutations. So it allows you to be able to sort, more. You could then narrow it down more specifically if you just wanted the variants that have a high degree, high likelihood of being deleterious. It can allow you to narrow down a little bit further. Can we export these tables? So some tables you can, yes. So the data exploration tables you can download the tables using an export as TSV button. Fortunately, we don't have that feature in the Variant Explorer yet. That is something that we are trying to add to the portal. That's next in our development. But we do support the Variant Workbench, which is a similar Chromatica-based tool where you can explore these tables in greater depth. Talk a little bit about that maybe on Friday. Seeing where the week takes us. All right, so I'm going to pause here. So this just kind of shows an overview of what Kids First is, and so you can see that we're really cross-disease, cross-condition organization, and so tying back to our Common Fund topics here, where we're if we're looking in each of these individual institutes, well, where would Kids First fit? Well, we have heart defect data, so would it sit in the the heart, lung, and blood? But we also have kidney data, which has its own NIH Kidney Institute. But there's also an NIH Pediatrics Institute as well, which also feels like a good spot for this. And so this is where this kind of common fund exists to kind of support these programs like Kids First and GTEx that it can support projects from multiple places. All right, and then I'm going to share one last example of a common fund program. So this is HubMap. This is the Human Biomolecular Atlas program. So their goal is to develop this open platform to map healthy cells in the human body. So they're really interested in looking at everything on the cellular level, whereas Kids First is really focused on the genetic level, and GTEx also looks at genetics, but they're very much at kind of a tissue-based level. HubMap is really kind of even finer detail here, and we're looking at specific cellular level, and a lot of their data is actually paired with microscope images of human tissues. So let's pause and we'll take a look at that as our last example here. So I'll open another window. We'll do NIH HubMap. And again, this is starting to look familiar, I hope. So now we're on an NIH website about HubMap, and it tells us details about what the program is. But if we want to learn more about HubMap, you can see that, of course, they have imaging, so they like to show off their beautiful images. But then we can visit the website. So this is HubMap. This is our third DCC that we're going to talk about. We're also going to access some HubMap data. As Sangeeta is going to talk a little bit about HubMap on Thursday as part of her single cell workflow. They have a nice overview video here, which talks a little bit more about what HubMap is. But we can also explore the different resources that are available. They have a new paper out in Nature where you can read more about it. So if we go into the data, and we go to the data portal, takes us to this site. So it has information about their data set. So they have 233 donors who donated over 2,000 samples. And so if we're interested in a specific tissue, so for example, if we do kidney, maybe we're interested in kidneys, it provides information about what types of data is available. So we have RNA-seq from kidney samples. We have a TAC-seq, so this is information related to epigenetics. It refers to kind of chromatin organization, identifying regions of the chromatin that's open. Could then suggest that you could find regions where transcription factors are binding, so helps you find transcriptionally active regions of the genome. LCMS, which is a type of metabolomics, so you're looking at mass spec. And then we have some autofluorescence and histology images. MALDI, which I believe is a proteomics technique, and slide sequencing. And from there, you can click on specific links, and so it provides information about these specific samples. So these files are those microscopy files. And I think if we click on it, we might be able to pull up specific information. So it provides the information about the specific sample. This one is not as pretty as the one on the front page, but it does provide you information about where these cells are coming from, and then linked to not just this picture, it also has the genomic and RNA data that's available and associated with this sample. So it allows you to be able to look at a higher resolution than you might be able to on the scale that Kids First does. And so then from here, it gives you information about where this sample came from. and then it provides information about, how to access this file, who were the researchers, and then who to attribute this to if you, use this as part of your research project, as well as if you're interested in doing some type of bulk data transfer, to be able to help identify files, to be able to use them. So this is, and the resolution improved too, so that's good. And so this is the hub map. So it provides, again, data on the cellular level. So if you're interested in kind of single cell, in order to kind of level to be able to learn a little bit further. Let's see how researchers are using these data to inform their research, how to best locate that information. Yeah, absolutely. So related to that, I would point you in a couple places. So a lot of these organizations have their own paper. And so if we go back to the hub map resources, they have tutorials and technical documentation in terms of how to literally use the data. But if you're interested in what other research projects folks are using the data for, I would check the citation and then see what papers are citing, the HubMap paper. I'll also do a call out on the Kids First side. If you click on our website, our Kids First website, we actually host a set of publications as well. And so these are all papers that were published that use Kids First data as part of their discoveries. And so if you're interested in reading about, well, what are folks using this for, you can scroll through here. You can click a particular paper and then view the article, and it will link you out to PubMed. So you can read more details about, the types of research as well. So great question. So yes, we're not just, kind of creating these data sets to look at them and appreciate them. Hopefully we're trying to get this out into the world and have folks use them as well. And so that's where we're highlighting here with these publications. Great question. All right. So I chose those three because those three programs are going to be part of our course this week. If I pull back, so that's our wrap up on HubMap. So each, like I said, this is going to be kind of the featured data sets that we're going to talk about. So on Wednesday, we're going to look at combining Kids First and GTEx together in Cavatica using an RNA-seq workflow. And then on Thursday, we're going to look at combining Kids First and HubMap together using a single-cell RNA-seq workflow. So we're kind of showing our different tooling there. So each of these is a program that's funded under this concept of the Common Fund. And so each program has its own goal, its own unique data sets, and its own unique platform. So Kids First, we're focused on these broad conditions. We have our genomic data sets. You find them on the portal. GTEx similarly has data from a lot of different tissues. Really interested in gene expression. It has its own platform as well. HubMap is very interested at the cellular level. They have their own platform, their own data sets. But you can imagine how data from each of these could help inform projects in other programs to improve research outcomes and discoveries. And so this was a question that I addressed a little bit earlier. I think Harun asked this question related to, well, how, what types of controls are available? Well, Kids First doesn't support controls, but we are interoperable with GTEx. And so you can combine the GTEx data with Kids First data to kind of look in specific detail there. And so you can see we want to still try to support and make it easy, us as Kids First DRC and working together as part of this organization, trying to, how do we get these groups to work together and make more valuable data sets and data resources that can support all these different projects. So ta-da, we made it. So after a long-winded preamble, hopefully you have an understanding, without even me having to say it, of what the Common Fund data ecosystem is. So the CFDE, it's the Common Fund data ecosystem, and our goal is to try to enable the broad usage of data from these different institutes to be able to accelerate discovery. So basically trying to build those little arrows there and support the creation of more of those arrows, across different platforms. And so the goal of CFDE is really to try to make these Common Fund data sets what's called FAIR. So FAIR data access, which is findable, accessible, interoperable, and reusable. So the goal here is that, exactly related to the question of how do we find this information? How do we locate the data files? So everything needs to be kind of findable. If you don't know it exists, then it's not doing folks any good. Accessible, related to folks need to be able to use it. Folks need to be able to work with it and touch it and interact with it. It needs to be accessible. Interoperable refers to being able to use these data sets across the different conditions and combine them together. So we're not trying to have to, start from scratch every single time and have to redo everything in Kids First just to get it to line up with the stuff in GTEx. We want everything to be seamlessly interoperable and usable across these different programs, and then reusable, such that what the findings that people make are reproducible, are scientifically valid, and they are driving the creation of knowledge in our world. So here we are, kind of a diagram here. So our CFDE is sitting at the center here of this web. And so I highlighted my three DCCs here with asterisks: HubMap, Kids First, and GTEx. So each program, Kids First program and the GTEx program and the HubMap program is its own data coordinating center. So Kids First is responsible for managing its own Kids First data projects. GTEx is responsible for managing its own data products. HubMap likewise, and you can see all these additional programs, which I didn't even get a chance to talk about today in my demo. So what CFDE does is it serves to coordinate all these DCCs together to try to make their data set and data products easier to combine. So get the folks in the same room talking together to be able to help support collaborative research. So let's take a step out of here, one last step out of here for today. And so let's look at the Common Fund data ecosystem site, because they have their own portal as well. So where did my window go? There it is. So I'm going to open another tab and we'll do NIH CFDE into Google, and it takes us, you already know where we're going to go. It takes us to an NIH page all about CFDE. So if you want to learn more about what CFDE is in detail, it provides information about it. CFDE has its own kind of centers that allow for different training and collaborative opportunities. And then we also have the CFDE portal. CFDE, this is the CFDE information portal, provides information about all the different Common Fund programs. And so if you're interested in kind of browsing this after the course, you'll be able to find the three that we talked about: Kids First, HubMap, and GTEx. But you can see that Common Fund programs really span this very broad set of different types of data. So if you're interested in microbiome, there's a human microbiome project. If you're interested in muscular development, there's this program called MotorPack. If you're interested in, I believe there's a kind of connecting human biology to model organisms, there's this called Comp2, which is looking at using mouse models to try to validate findings in human genomes. 4DN is entirely based on the organization of the genome in 3D. So lots of high-C data in terms of kind of genomic structure, three-dimensional structure within the nucleus. Again, going back to information related to the accessibility of the genome and the DNA molecules to these transcription factors. And each of these sites, you can expand and read more about it. And then you can also, it has links out to their particular portals as well. So you can explore, sites that we're not going to get into in the realm of our course, but it does provide some overview in terms of, other types of CFDE data that's out there. It also has opportunities, provides different training opportunities. Hey, that's the one that we're at. So we're at NIH Kids First CFDE Pediatric Power Up. This is the second one. We taught this in June, and this is the August one. But you can also see other training events as well. So for example, at the end of September, HubMap is hosting their own event where they're diving deeper into the HubMap data. So you can kind of explore different data sets and see other types of outreach opportunities if you're interested in learning more about the data sets in the future. Let's see. And then if we go onto the data portal, it provides information about our CFDE data portal. So it lets you search specific metadata, specific process data sets. So for example, if we enter a keyword, maybe we try stat3 because that was recommended to us. So this gives us information about this gene called stat3. And so it kind of links out to all these different sites. And so it tells us about expression of stat3 in these specific disease conditions. And that comes from the links data set. We can scroll ahead. Lots of links data. Maybe we scroll by 100 at a time. So lots of links data related to gene expression in different conditions. There's glygen, so this is related to glycosylation. So it looks at in both human, and there's also some model data there as well. And so it provides you kind of an overview of different places you can go to access more information related to your particular gene. So it gives you kind of a starting point to be able to search and explore. So the GTEx workbench, if we go back to search, maybe we're interested in kidney development, so we could type kidney. So it gives you information about, kind of this concept of kidneys, but then it also has specific kidney disease conditions. It gives you information about, gene expression. So there's that kidney data from GTEx. It gives you information from MotorPack. So this is from rats from a MotorPack study related to kidneys. And so it provides you all different types of information related to kidney. So it really gives you kind of a good kind of starting point to explore, say, what is out there. This is my gene, this is my condition, what types of stuff are there. From that kind of launching point, it says, oh, there's data about kidneys in GTEx. Maybe I should check this out. And it links out to the GTEx website. So that's our overview of CFDE Workbench and how to kind of use it to get a sense of what's out there, what types of conditions, and what types of genes that you can search for. All right, so now I'm going to go back in here, and I'm going to pull up this slide. And I believe this is my last formal slide, and then I will share where we're going to go next. All right. So that was my conclusion of my kind of content for day one, but we are going to continue here in a little bit. And so we've talked about what is CFDE, hopefully provide a good overview of where we're going with the rest of the course. We talked about this concept of what a DCC, a data coordinating center, is, and we looked at 3 of them that we're going to explore later on as well on Wednesday and Thursday. We looked at the different types of data sets that these DCCs make available, and we showed how you can begin to kind of access these data and search for them on the CFDE portal. For now, I'm going to highlight, so on Wednesday and Thursday, we're going to do these analysis walkthroughs. So we're going to be looking at doing our own analysis. And specifically, we're going to be using analysis in this thing called Cavatica. So what is that? Let's learn a little bit more about that, and let's set up today so that we will be ready Wednesday and Thursday when those roll around. We'll be ready to kind of dive in and get started. All right, so what is Cavatica? So let me pull up my internet browser here. And they actually have a really good front page, cavatica.org. So what is Cavatica? When we talk about doing analysis, these kind of genomic level analysis, we have these very big data files, and we need to identify information inside of them. So you can imagine if we have an alignment, we need to find maybe all the variants in that particular sample that are different than a reference genome, with the thought that some of those variants might be responsible for the underlying or the condition that the patient, or the participant is diagnosed with. So if we have a genome of a participant with a heart defect, maybe we would want to identify the genomic variants in that individual and then try to identify which one of those might be involved in heart development to be able to impact this participant. So in order to be able to do that, you can imagine we have this huge text file of all A's, C's, G's, and T's. And so you can imagine scrolling through that and then comparing it to another file and scrolling through that and trying to find, where the A's and C's and G's and T's are different. So that would be not a very good way to do it. It would take you forever. You'd probably miss some things. So we use computers to help us. So computers are really good at searching and finding and comparing and highlighting differences in different large collections of text. And so we can use that type of tooling to be able to help us. Up until the last couple of years, the very kind of beginning of genomics, a lot of that work was done, what's called in the computational world, done locally or on-prem, on the premises. So what that means is that, as today, individual institutions have their own high performance computing cluster, their own resources. And so if you work at Harvard, you have access to Harvard's cluster. If you work at Hopkins, you have Hopkins, and so on. Depending on what institution you're at, your institution supports its own, very powerful computer, which is really good at doing this high power stuff in order to be able to access. So there are some caveats about that, in that one, you have to work at these places in order to be able to use these computers. And so if I don't work at Harvard or I don't work at Hopkins, but I still want to do this genomics, well, how do I get involved? How do I do it? And then likewise, a similar caveat is, well, what if I work at one of these places, but my collaborator, works at UCSF across the country. And so I can't give her access to my data because my institution doesn't like it. And, I can't access hers because her institution doesn't like me. And so how can we kind of, even though, obviously we want to work collaboratively here. So enter the proposed solution, which is what's called cloud computing. And so what cloud computing is we're doing this similar high-powered, high-performance computing, but we are borrowing someone else's computer up in the cloud, so to speak. It's not literally in the clouds. It's actually at a data center, probably in, around the world, maybe Virginia or Ohio, if you're in the Eastern United States. But, we're borrowing someone's computer just for a moment, while we're using, doing our analysis, and then we're, letting it go back to the folks. So it's kind of like a shared resource that allows us to be able to do this type of computing without having access. Again, if we don't have access to a specific institution, again, or the other advantage is that it really allows for easy sharing across different institutions and folks around the world as well. So that's the concept of cloud computing, and it's the same concept. Maybe on your phone you might have a cloud storage for if you have lots of pictures. So you're using the storage space of someone else's computer to back up all your pictures. So this is a similar concept, but we're using it for genomic analysis. Okay. So then what is Cavatica? So Cavatica is a cloud storage analysis and sharing platform. And so what this is basically, it's kind of a way to be able to access and interact with those powerful computers without having to be, an expert, without having to, know all the ins and outs of Amazon Web Service and Google Cloud Platform and all of the different, kind of messy details of computer science. So they've kind of set up the system for you, and we're able to log in and work on the platform. So related to cloud computing, so Cavatica platform is free to log in. It's free to create an account, and it's free to explore. But once we get to the point where we want to run an analysis, where we're borrowing the computer, so to speak, it does charge us money. So it does cost money to borrow the computer, but the costs are relatively small compared to the upstart costs of buying and setting up and maintaining a massive stack of computer servers in your own institution. So it allows you the time to focus on the genetics and the genomics and the scientific discoveries, and less on the kind of computational cluster building side of scientific research. So that's my preamble to what cloud computing is and what kind of services Cavatica offers. So we're going to get more in depth on Cavatica on Wednesday and Thursday. We're going to dive in here in much more details, and I believe we're going to have some folks who help build Cavatica as part of their support team. They're going to join us and help us as well in order to be able to answer questions. And so this site is cavatica.org. It gives you information about what Cavatica is. So Cavatica is specifically related to pediatric tumor genomics data, but I would broaden that to even say pediatric genomics data, because it does allow us to be able to access our birth defect data sets as well. Produced by this collaboration with this company called Seven Bridges, based on their thing called the Seven Bridges platform for cloud storage. And so this Cavatica page, cavatica.org, it gives you kind of overview information about what Cavatica is. It talks about, how do you get data in here? So there we are in Kids First DRC, and there we are in Common Fund Data Ecosystem. And so it talks about, ways, kind of folks that are using Cavatica as a platform. And they also have a great set of getting started pages and tutorials as well. So how do we get started here? how do we kind of, set up and onboard. So they have a link, mix of videos as well as written content, as well as kind of documentation as well. All right, so how do we get started? So you can kind of explore the knowledge on your own, but I'm going to demonstrate here at the front how to log in and create your account. So you log in, and I think it probably links from this page. I imagine if we go to the home page, it links at this very first word, Cavatica. Will take you to the website. Oops. Cavatica.sbgenomics.com. And so that will take you to this page. And so it gives you two opportunities to log in or to create an account. So the first is if you have an NIH ERA Commons ID, you can log in just with that. And if you click this button, it will take you to the NIH page where you can log in with your NIH ERA username and password and click sign in. If you don't have one of those, that's okay. You can create your own account with any username and password using any email. So for example, if you take it here, it asks for my username, and then which I have access to our kind of our admin account, and then our password, and then I can log in that way as well. So it gives you two kind of opportunities. If you're new, this is your first time, then you need to create an account. And it gives you those same two options. You can create an account with your ERA Commons, or you can create an account with an email and password. Once you do that, if I click the second one, the email and password, it gives you, some basic information about who you are, email address and a username and a password, and some organization. At this point, then it asks you to, agree to the terms and services and confirm that you're a human being, and then allow you to click the register button. So it takes a very kind of basic setup there to help you get started, create an account. So once you create an account or you log in, I'll go back and I'm going to log in with my ERA Commons ID. You should see a page. It looks a little similar to this, but it's not exactly like this because I probably have a lot more things on my homepage than maybe you do. So on the left-hand side, it describes this concept of projects. And then on the right-hand side, it has this concept of different analyses. So we're going to dive deeper. I think Sangeeta is going to provide a little bit more instruction about, what these are and how this sets up. And definitely on Wednesday and Thursday, we're going to, show different examples of, what our different projects are, how do we create a project, and how do we run analyses. So that's what we're going to look for, moving ahead. But the last thing I'll show you is that there's a way to get help, and that's this blue question mark arrow down here at the bottom. And so this allows you to be able to get help from the Cavatica team to be able to answer specific questions. And so one thing I'll highlight, so I mentioned that Cavatica offers, it's free to sign up and it's free to create an account. But once we start running, using the computers, then it costs money. The Cavatica actually supports a $100 kind of startup pilot credits that allows you to be able to get started, explore, and it's definitely enough for this course that we're working on, and we'll take you even a little bit further beyond the course. And on Friday, we can talk about, where do you go after your $100? Turns out there's lots of NIH resources for getting additional cloud credits to support, projects in the cloud. But you can contact their support in order to unlock that $100. It's kind of hidden here. But if you click this blue question mark down at the bottom, and you can type in this box, you can say, hi, my name is such and such, David. And it's going to capture your username. It's going to capture you're on Cavatica when it sends this message to the Cavatica support team. And you can say, I am a member of the CFDE Pediatric Power Up course. I am interested in getting started and accessing the $100 pilot funds. Thanks so much. Those will be polite. And so what that does is that we'll send, and then you click the send button. So what this does is this confirms that you're a human being, you are interested in using the platform by sharing that you're associated with our Pediatric Power Up course, then the Valsera team will recognize you and hope to get you set up with those pilot funds. So at this point, this is just kind of a short little intro, and then you can click the send button, and then that will automatically send an email to the Convatica team. The other way to do it is you can also write them an email, support@valsera.com. And if you do that, then you would want to include the fact that you're working on Convatica, your Convatica username. You would want to mention the course, CFDE Power Up, and you would want to mention the $100 pilot credits. So if you choose the email route, then that's kind of the short list of information you'd be looking for. And your Convatica username is linked up here in this dropdown. So my username is HigginsD, but yours is part of that dropdown. All right. So what that does, hopefully that will give, if we do this today, hopefully that will give the folks behind the scenes time to set these up. And then when we get to the course on Wednesday, be able to follow along, not just by watching, but also by, on the keyboard as well. So I wanted to highlight that. All right. So I think that covers everything that I wanted to cover today. Like I said, not all the courses are going to go the full 2 hours. And today especially, we're going to wrap up early, but tomorrow will probably be a little bit longer. So just kind of a forewarning for that. So with that, I'm going to stop sharing my screen. I'm going to say thank you all for joining today. It was great to meet all of you as briefly as we could through the chat. But we are going to pick it up again here tomorrow, where we learn about different bioinformatic techniques. So today we learned about the data sites. And then when we return, we're going to learn about how do we actually use those data sets, how do we get information out of them. And so Sangeeta is going to tell us all about that tomorrow. So with that, I'm going to close the session today. If you have questions, then feel free to stay on a little bit later. Sangeeta and I will be available to answer any questions. We'll stay for a couple minutes at the end of course. And then otherwise, we'll see you tomorrow at the same time as we did. So thanks so much for joining, everybody.